today I really want to share some of our efforts on building highly specialized, but hopefully towards the generally scientific agents that really focus on how can we program genome and cells as cures for a lot of disease that we believe will be highly impactful for aging, for health, for treating disease. So this is our group. We have a mixture of computer scientists as well as wet lab experimentalists that are working together in the group building AI agents and models. So briefly, we have three parts of the lab. The first two parts focus more on wet lab discovery, building gene cell editing tools, how to program immune and stem cells. And today I want to focus more on the third pillar of our lab, which is building machine learning and AI approaches for exciting medicines. And my thought is really, as most of us know, right, biology is very the slowest, most painful experimental scientist field, like we've heard from the speakers earlier. And then even though we have computers, internet, I think the progress of pace of discovery in biology, in medicine, has largely stayed the same. I do believe, as Sam was saying, we have an amazing opportunity ahead to take that from a linear, slow sort of growth into exponential explosion. And this is why we're really excited about using AI models and agents. But the problem is, does AI really understand, or at least the current general purpose AI really understand biology? We believe, as really shown by multiple speakers today, is very hard. Because general purpose LM usually have still problems with hallucination, and we are talking about life-saving medicine. It's really difficult to believe in something that might hallucinate a protein or RNA that doesn't actually work, right? And then there are always a lot of problems giving too many options. But in really scientific research, we need a very specific, grounded, explainable solution, rather than a bunch of options or maybe an exam-like answer, right? So, and finally, we think we need to put agents into real action, validating lab, generating these cycles, as we all sort of agree. So our approach to thinking about how to build AI sort of workflows for biomedical innovation is sort of laid out here in sort of a acronym ABC. So A is sort of the interface, which is we think that AI agents will really work with human scientists in a variety of biomedical innovation tasks. But the agents are building on top of a foundation of biological aware, biological specific models, like protein language models. We have been working on RNA language models and other type of multimodality models behind the agent. And finally, to really push the whole field forward, we really believe in the value of getting benchmarks, standards that are setting up across different tasks, complex sort of scientific discovery tasks, as pioneered by many groups, including Filter House, right, as we're studying from Lab Bench. So from these agents, models, and benchmarks, I think we can really push the whole field forward. So I want to touch on three components briefly today in my talk. So starting actually, it may be backward from the benchmark, which I actually think is really critical, even for building an agent that actually we know works. We've been building specific benchmarks such as Genome Bench, where we really take real-world discussion, not from a paper or maybe a random sort of a clip on internet. It's really from real genome scientists in their actual research process. They were discussing science, troubleshooting experiments, and this is thanks to our collaborators over 11 years of collection. We got this benchmark set up, distilled from real human experts when they're actually doing research. And the result of that is we can actually use this benchmark to build really strong reinforcement learning approach, shown here, which we call RL router, meaning that because the ability of different models to perform really well at different tasks, let's say some model might be really good at designing RNA or proteins, some model might be really good at experimental troubleshooting, we realize that we might need two layers of RL, right? You have the typical RL focused on the model, but maybe we can have the RL over RL where we really understand what models are really great at doing specific tasks. Integrate almost like a MOE type of approach where we can raise a router, almost like an open router approach, where we can route the question, the challenging questions, to the best model answers to the task. You can see that the RL router at the bottom really outperforms any of the best reasoning models in this genome bench. So I think this towards building a specialized really potent, our approach for science is going to be greatly accelerating our work in building agents. But it needs to start with a benchmark. And then building on this work, we've started to build agents such as CRISPR-GPT, which was published earlier this week, where we can really use that knowledge, for example, in this case, from genome biology, genomic gene editing, to really improve our performance for anything that related to CRISPR gene editing. And you can see that it outperforms a lot of the best models that we tested. I'll give you an example of how this would work with human. You can see that we can have a really nice interface where any scientist doesn't need to know how to code, how to do specialized analysis, can just use natural language, interact with the agent. And you can basically lay out your task, and the agent really understands the whole context of your own project, your lab, that really can help you build a workflow, as you can see on the right, based on the need and the context of your own research. So as you understand better of your research, you can really start building a complex workflow, as shown here, that help you to do design, analysis, and all the things that you wanted, for a specific domain task like gene editing. And we think that this will be very enabling for people because you no longer need, for example, the PI to be involved, or you need to find some expert. Anybody can really use the agent, knowing that this is built from 11 years of expert knowledge and know-how. So this is what we think a power is to really scale biological scientific discovery. And so, of course, we want to validate it in real lab. So since this is just published, I won't go into detail, but basically we validated using, for example, a first-year college student that on the first goal of doing genetics experiment, they can achieve 80 to 90% success. We were really happy that on the very first test, everything basically works. And really thanks to AI agents, and there's really no involvement from the professors or the senior postdocs. And building on that, very quickly, we also have been expanding the effort beyond just gene editing. Recently, we'll be working with Dr. Matthew Porteous, one of the leader of stem cell biology and gene therapy. As you know, Dr. Porteous' work is really behind the first approved CRISPR therapy by FDA, Casgevy, and also Graphite, which is a public company working on stem cell engineering. So we really think that working with the leading expert in the field, we can build very potent specialized agents like stem cell GPT here. This is not yet released, but we're hoping to really validate this agent and release it very soon. It's basically building on top of the work we did before, really give you an expert that help you with everything about stem cell engineering, particular focus on hematopoietic stem cell and induced pluripotent stem cell, iPSCs. Give you one example. The agent actually can design personalized editing strategy based on, for example, SNPs. As you know, all of us share a lot of DNA sequence, but each of us have some small changes called SNPs that are different. So if I only design a general CRISPR or engineering task, it may not work for another person because of small genetic difference. So to tailor, customize the strategy of engineering stem cell for each person is really a lot of work for human to do. But with the agent, now we can really say it's almost fully automated because it takes in personalized genomic information and designs strategy based on, for example, the donor's profile for that donor's stem cell. So this is really, we see how we can scale almost any domain-specific, extremely labor-intensive task using agentic workflow, as shown here for stem cells. And finally, one last part is I want to show you that we are very interested in bringing this agentic workflow to a broader scope of tasks in biomedicine. And one of the work that's already been pre-printed and really exciting to have a lot of traction already is the Biomni, which is open source work really led by Yuri Ashkova's group at Stanford CS, but we were collaborators along with Aviv Regav at Roche Genentech, is more towards a generalist platform for building these agents for a lot of different tasks. And then we also will be releasing a functional genomics agent called AutoScreen very soon, where we believe that we can use this agent to perform any type of target discovery functional screening, for example, like CRISPR screens, human genetic GWAS studies, where we can really build a lot of agentic workflow for a variety of tasks beyond just the CRISPR and stem cell. And finally, one last thing I want to mention is that we have found that, as you know, why don't we let AI evolve themselves and try to improve themselves? Like Sam were just talking about, right? Why don't the AI be able to, at some point, get smarter, almost like a real human, right? Learning themselves, almost like an AlphaZero. type of approach. So we've been building self-evolving agent like Stella, which is just pre-printed recently with Dr. Mengdi Wang at Princeton, where we really are thinking about if we give the agent access to a lot of tool and approaches, APIs, and very defined benchmarks and tasks, can an agent really evolve their own ability to further improve, almost doing a self-RL. And we found that, for example, even without much fine-tuning or specialized design, Stella can be a lot of the frontier model like Gemini, even Bellman in some cases, using benchmark, really thanks to Future House's work here. So as basically as we've seen, finally, when we have agents self-evolving, we can now give them more data. So we have been actually using SR glasses and cameras to let the agent see what is actually happening in the real lab. And we can see that the agent can now, using VLM, really empower people to understand and guide their lab experiment in real world, rather than just on your computer. So we really believe that this will open up a lot of opportunities. And sorry, I'm a little over time, but one last thing is we do have a concern that how this powerful agent will really impact real life. So we're working on, we just have announced the first biosecurity workshop at Europe this year, and we're really looking for people to be mindful as we're building powerful tools, the security guardrails that we need for dissemination. Thank you very much.